Our next speaker is Sophia Dasaro. She's a graduate student at Purdue University, and her presentation is about peptide delivery and nanoparticles, two hot topics around for the past decade. So we appreciate to know more about your work.
Awesome. Thank you for the introduction, and before I get started, I'd like to extend a huge thank you to Simon and our sponsors for helping make this possible and giving me the opportunity to speak about the wonderful research our lab group does, and I'll be continuing into my PhD. So, as our chairman said, my talk today is focused around our lab group's technique for encapsulating peptides into polymeric nanoparticles. So it's no secret that there's industry interest in biologics. They're a rapidly growing area of interest, and that's because of all of these advantages associated with these molecules. They're highly specific to their targets, and they can be engineered to interact with these specific targets. This has given way to novel treatment strategies for many, many challenging to treat, previously untreatable illnesses. However, these biologics can be challenging to deliver. If they were easy to deliver on their own, a lot of us wouldn't be in this room or with a job. So our group has identified that encapsulating these biologics into core shell nanoparticles might be one strategy to help deliver these life-saving molecules intact to their sites of action. Our group's technique that we use to encapsulate molecules is called flash nanoprecipitation. This technique was developed right down the road at Princeton University in Bob Prudhomme's lab. Flash nanoprecipitation is a scalable anti-solvent precipitation technique that is originally designed to encapsulate strongly hydrophobic drugs. This technique works using specialized mixers with specialized geometries. This is a confined impinging jet mixer, and for those of you out there with a bit of fluid mechanics background, yes, it's true that the mixing plane does not look like this in an impingement jet mixer, but for the sake of the cartoon, I've illustrated it this way. So once the streams are combined in the mixing chamber, on the scale of tens of milliseconds, nanoparticle assembly takes place, starting with the precipitation of your active and stabilizer. The operating principles for this technique are a short timescale to achieve homogeneous mixing. By this I mean that the inlet streams are rapidly combined and homogenized before the components start to precipitate. This is because the particles form via diffusion-limited aggregation. And if the timescales of diffusion are mismatched for your components, you're not going to get a homogeneous particle size distribution. And then with that, to complement that, the second operating principle is the fact that your components need to have similar timescales of precipitation. If your active precipitates before your stabilizer, you're not going to encapsulate anything. So advantages of this technique are that it allows for this core shell architecture, right? You can tune that hydrophilic block of your polymer to achieve pH responsiveness, temperature responsiveness, as well as improve interaction and cellular uptake. It allows for this modular approach to formulation design. Another advantage that's not listed on the slide is that in the lab, we can do this at relatively small scales, which allows for high throughput screening of molecules in the cases where, and there often are, you don't have a lot of drug to work with. It also has high encapsulation efficiencies and slots right into a continuous and reproducible manufacturing scale. So the key limitation of this is that it works and was originally designed for strongly hydrophobic drugs. Biologics, peptides, proteins, and mRNA are not strongly hydrophobic. So how do we overcome this? We can combine flash nanoprecipitation with in situ hydrophobic ion pairing. Hydrophobic ion pairing is a solubility modulating technique that forms a water-insoluble complex between your water-soluble or weakly hydrophobic active and a hydrophobic counterion. Advantages of this is now we can encapsulate weakly hydrophobic drugs as well as hydrophilic molecules. It increases encapsulation efficiency further, and it doesn't actually alter the physical chemistry of your active. This is to say that once those ionic bonds are disrupted in the body, your active returns to its original state. Some cons to this are that it reduces your drug loading by mass, right? Because now for every molecule of your active, depending on the number of charges, you have one or multiple parts of another excipient. However, it does allow us to encapsulate water-soluble or weakly hydrophobic molecules, which we previously couldn't with this technique. So now's a good time to mention and segue into the motivation for this research project. So this process of combining a precipitation technique and hydrophobic ion pairing is not unlike what we use to encapsulate mRNA for lipid nanoparticles, but it's not the same. Because unlike RNA, peptides and proteins can be heterogeneous in their structure and charge. And RNA has this repetitive charge and is uniformly hydrophilic. So the motivation for this work was to better understand how these techniques work to encapsulate peptides and proteins. So in this work, we selected polymyxin B as our model peptide. This is because it is structurally complex, it has 5 charges, and as well as a balance between hydrophilic and a little hydrophobic domain at the end there with that aliphatic tail. So we've selected this peptide, right? And our objective here is to demonstrate that we can use these combined techniques to encapsulate this peptide for a suite of different hydrophobic counterions. In all cases, the hydrophobic counterions have this similar structure. They're all surfactants. And specific goals for this project was to demonstrate, as I said, the feasibility of this technique, and as well as figure out how this encapsulation process affects particle assembly and drug release, and then hopefully translate this mechanistic understanding to controlled release formulations for other peptides and proteins. So before I step into the results, I'd like to walk through how that nanoparticle assembly process changes when we include the hydrophobic ion pairing step. And the answer is it doesn't really change much. We just have this ion pairing process after the streams combined. So for all of the data I'm going to present, in our solvent stream, we had a hydrophobic counterion, which varied, and then our stabilizer was fixed to PCl-PEG. But truthfully, for the FMP process, you just need something that has amphiphilic character. We've used HPMCAS in previous experiments, and any diblock copolymer will work. And our particles are stabilized, in this case, through steric interactions via those PEG blocks. But in other cases, if that outer block has a charge, then you have electrostatic stabilization. So there's flexibility that way. We also found, unique to this work, that this ion pairing process results in liquid crystalline phases forming in the cores of our nanoparticles. This is unique to the work that we've done with the ion pairing process between polymyxin B and a suite of hydrophobic counterions. And this was really interesting. But how did we figure this out? We used synchrotron small X-ray scattering, or SAX. So ordered systems, as we've seen in many of the presentations, will scatter X-rays and generate patterns. These patterns, in this case, in this work, were mathematically translated to these 2D patterns, and then we use the relative ratio of these peaks to identify which liquid crystalline phases were exhibited in the cores of our nanoparticles. So the first thing that we varied in this work was the charge ratio. That is how many counter ions per charge on our cyclic polypeptide. Polymyxin B has 5 positive charges, so at a 1-to-1 charge ratio, we had 5 counterion molecules. And we varied this 1-to-1 and 1-to-4. And we did not find that increasing the charge ratio drove a change in LC phase. What did, however, was the media. So now that we know that we can modulate the liquid crystalline phase as a function of formulations or aqueous environment, we extended our counterion suite. And so what we did in this series of experiments is we fixed the length of that carbon tail to 12. And we varied our head group geometry. So we changed out that counterion for a whole bunch of different counterions with different geometries. And we find that there are different liquid crystalline structures associated with these different counterion geometries. We explain this loosely in using the critical packing parameter. Another relevant term to this is the hydrophilic fraction. These things are somewhat interchangeable, but both of them use the relative geometry of your amphiphile to predict how it will pack at an interface. So in general, as your critical packing parameter increases, we see the negative curvature of the system increasing and an increase in internal ordering. But how does all of this play into the counterions we've selected in our screen? Well, the carboxylic acid, compared to the sulfonic acid, has a higher head group area, therefore lowering the critical packing parameter, bearing all else the same, and therefore should be less ordered than the sulfonic acid head group, which has a lower head group area and therefore comparatively high critical packing parameter. So all of this was really interesting. We can switch out these counterions, play with the charge ratio in the aqueous environment, and get a whole suite of different liquid crystalline phases. But this is a controlled release summit, so it's important to focus on the performance of these nanoparticles. And we found that there is, in fact, a difference in nanoparticle performance, which we associate with LC phase. In general, and well, not in general, but looping in all of the experiments done in this work, we find a correlation between the orderedness of a system and relative drug release. So in the plot on the right, we have the same active at the same concentration in all of our formulations. The only thing that's changing in that plot is either the concentration of counterion, so how many charges per charge of our active, and the active geometry. And we find that for polymyxin B, which is a highly water-soluble peptide, over 48 hours, we can achieve anywhere from like 15% release to up to 100%. And this was really, really interesting. And we also found that these trends hold in release media pH. So at PBS, at pH 2.3 as well as 7.4. So all of that in vitro data, well, in vitro lab bench data was great. So the next thing that we looked at was how does this perform in an animal model? So we selected a bacterial lung infection mouse model, and we dosed intratracheally. We did this in collaboration with a group at Monash University in Melbourne, so I didn't do any of the animal work, but we did find that the nanoparticle formulations resulted in a higher, a more efficacious than the free drug against this lung model. So far in the presentation, I've demonstrated that FNP works for hydrophobic drugs. That's how it was developed. And we also demonstrated that FNP works with hydrophobic ion pairing to encapsulate hydrophilic or weakly hydrophobic drugs. Wouldn't it be awesome if we could co-encapsulate these two things together to achieve a synergistic effect between both actives? So an example of this would be a vaccine antigen and adjuvant. And this is the focus of my thesis work, which I'm currently about to enter the third year of. So this works because FNP relies on hydrophobicity. And when we combine FNP with HIP, everything is hydrophobic or becomes temporarily hydrophobic during the process. This facilitates the co-encapsulation of hydrophilic or weakly hydrophobic molecules, so long as they have an ionizable group, and strongly hydrophobic drugs. But if we want to do this and have it be therapeutically relevant, we have to understand not only how this affects the nanoparticle assembly process, but how it affects the relative efficacy of both actives involved in the process. And step one is identifying how these two actives intercalate with one another in the course of our particles, to develop a really robust mechanistic understanding, because this remains to be known in this field. So this is a cartoon, right? Lamellar phases aren't these perfect, neat sheets always. So this is just a schematic for the sake of conveying the point. But we don't really know how these other strongly hydrophobic actives would integrate into the ion-paired biologic system. So one hypothesis is this phase separation, right? Our hydrophobic thing associates more closely with itself than it does with these lamellar phases by our ion-paired complexes. Another possibility is that this strongly hydrophobic API wants to integrate into those lamellar sheets. And then the third and least likely scenario is something that I just kind of brainstormed based on the nanoparticle assembly process, and that is that this hydrophobic thing is going to want to move towards that hydrophilic block of our copolymer. How are we going to figure this out? SACs again. So we can use small angle X-ray scattering data to actually delineate the thickness of these lamellar phases, and we can look at how that repeat distance changes. I use lamellar phase in this example, but we can use SACs to actually parse out the spacing of the other phases if formulations generate other liquid crystalline phases as well. And so putting that all together, here's an example of my most recent data set collected last summer. So we have a control nanoparticle, right? No hydrophobic co-API. This is just our ion-paired biologic. In the case of phase separation or surface association, you'll notice that the lamellar sheets are undisturbed. The hydrophobic thing would rather associate with itself than intercalate in these bilayers. And so we expect to see an overlap of that primary peak. This is, the figure on the right is zoomed in on that first peak of the scattering pattern. However, when we co-encapsulate with a material that integrates into the lamellar bilayers, we actually see a peak shift towards lower Q. There's an inverse relationship between Q and spacing, so what this means is those lamellar sheets are actually getting farther apart. And so how will we parse out between the phase separation or surface association case? Well, we're just going to do release tests, because we hypothesize that something that is associated at the surface is going to delay the release of the hydrophilic peptide even slower. And so with that, I'd like to acknowledge my lab group and our collaborators, because without them, I wouldn't be here, and I really appreciate their training and their belief in me as a second-year PhD student to come give this talk. And I'm going to finish off with just this slide on our group's active research areas. I'm obviously in the top two, but I can speak a bit to the third one. And as far as plant biology goes, I will plead the fifth, but I'm happy to talk about the work that they do the best that I can. Oh, yeah,
Sophia,
questions.
any questions?
Hi, Sophia. Nice talk. I have 3 questions for you.
Okay.
So, what sort of encapsulation efficiency were you able to obtain with your formulations?
Great question.
Yeah.
So it actually trends with the amount of counterion, but we're talking upwards of 90% encapsulation efficiency. Validated via HPLC, we collect our particles, spin them over a filter, free drug comes through, and we put that right over HPLC and quantify the amount of drug not in our particle.
Thanks. Have you studied the effect of your formulation process on the secondary and tertiary structures of the proteins?
We haven't moved on to proteins yet, so no, but I know that I think Bob Prudhomme's group has published a bit of literature on that, encapsulating proteins or effort done to encapsulate proteins.
And the last question is, if ever you need to lyophilize your formulations, have you studied that part?
I haven't studied for this specific encapsulation of peptides, but I have made polymeric nanoparticles through this process that I explained and spray dried them and also lyophilized them before. So it is possible with this architecture, but obviously you'd have to tune the lyophilization process in your matrix former to the composition of the particle and as well as the properties of your active. But it is possible. They have done that before.
And have you seen any effect on the performance of your formulation of that, for that matter?
Like post-lyophilization process? Not me personally, but in the work where I spray dried my particles, we were able to get identical DLS traces before and after spray drying. So take the spray dried powder, resuspend it at the concentration we spray dried it, and we see this really beautiful agreement. So we do have parameters in place in our lab to ensure redispersibility and all of that if we move down that route.
Thank you.
Any other questions?